Welcome to Life in Biology. This is episode 18, and as we prepare for March Madness, I'm going to title this video Single Cell Bracket Busters. This is a follow-up to episode 16 that was cut off right at the 10-minute mark, and this was the figure I was going to show next. And in the episode 16, we talked about hematopoietic stem cells and how if you take a mouse and lethally irradiate it, and then later inject these hematopoietic stem cells into it, the mouse's entire immune system, all the different cell types of the immune system, get reconstituted. So then that led to people trying to decide what is the order of differentiation as you go from hematopoietic stem cell to all these different fully differentiated cells. And so they came up with this model, and it's the first thing that my immunology students were faced with this year when we were going through our textbook, and it shows that hematopoietic stem cells are long-term stem cells, can renew themselves. It's a self-renewing population. It can be it can go on to be short-term hematopoietic stem cells and then multipotent progenitors. And then the first division towards differentiation towards one cell type versus another occurs when you have common lymphoid progenitors go off in this direction and common myeloid progenitors go off in this direction. So the common lymphoid progenitor can go through subsequent rounds of differentiation and become B cells, T cells, NK cells, and the book points out dendritic cells. And then on the other side of this, we've got common myeloid progenitors that can go on to be different cell types, neutrophils, monocytes, and I don't have it drawn here, but monocytes can go on to be macrophage-like or dendritic cell-like, eosinophils, basophils, megakaryocytes that break apart into platelets, and then erythrocytes, which is another word for red blood cells. So from this common myeloid progenitor, the model says that you should be able to end up with all these different cell types. In that episode 16, we also talked about how once you lethally irradiated mice, you could either inject the total bone marrow cells that hadn't been fractionated, and you'd need 100,000 cells to fully recover the immune system. However, they worked it out so that if you found cells that were lineage negative, which means that they don't express any of the markers of a differentiated white blood cell, SCA1 positive and c-KIT positive, also known as LSK cells, if you inject these, you need downwards maybe only about 10 cells to inject into that lethally irradiated mouse to recover the immune system. So this is the phenotype that people use to define hematopoietic stem cells. CMPs, common myeloid progenitors, have a similar strategy for defining them. And it's really quite similar to the hematopoietic stem cells, but here you have lineage negative still, so they remain lineage negative. You have SCA1 going away, it's now gone. You had SCA1 there with hematopoietic stem cells, and now it's gone. And then there's c-KIT positive, so that's the same as the hematopoietic stem cells. So the idea is if you select cells with this phenotype and inject those into a mouse that's been lethally irradiated, or if you just let them differentiate in something called colony-forming cell assays, then you end up with a complete myeloid cell department. And it can be myeloid cells of all sorts of different types, and I've listed them there, but we've already gone through them over here where that CMP branches off into all these different cell types. So the old school method, like I mentioned, was to just take these cells that have this lineage negative, SCA1 negative, c-KIT positive combination and do things like colony forming cell assays. So just real quick, what this assay is that once you isolate these cells, you put them in some media that's been thickened with methyl cellulose, and the methyl cellulose will prevent the cells from moving around, so that as your cells start dividing and becoming other cell type and becoming more differentiated, each place in your tissue culture dish where you had a single cell to begin with, you get a bundle of cells in one area, and they don't go drift over and mess up a bundle of cells in another area. And then what they determined from these sorts of assays, or what they suggested from these assays, is that this combination can turn into all those different myeloid cells. So that's the basis for this common myeloid progenitor going into all those different cell types. So now there are 3 studies that I'll link to below that had new experimental had some interesting experimental designs that go on to say that the old view is not quite right anymore, busting the brackets. So one is to barcode each cell. So in this method, you take a bunch of CMPs, quote unquote CMPs, and you infect them with a low number of virus, and then a small percentage of your cells are going to be infected with the virus, and each virus is going to inject a specific sequence of DNA that's different from what the other viruses are injecting. So in this way, you can keep track of a single cell. So if you go on to do the colony forming cell assays, or if you go on to inject those in mice, you can later collect the immune cells and look for this DNA barcode that the cells have in their genome, and you can determine whether, say, if a common myeloid progenitor is true and it can become all those cell types, you should be able to find this barcode in all of those different different sorts of differentiated white blood cells that are supposed to come from the common myeloid progenitor. So that was one experiment. Another experiment is to use more markers. So this is just 3 markers. Cells express plenty more proteins on their surface that you can keep track of. So rather than just using this set of 3 markers, and to be fair, Lineage is more than one set of markers because it's the absence of any of the lineages, but a limited set of markers, and they went on to use even more markers than what was necessary to get this the CMP phenotype according to these rules. And then the third experiment was single-cell transcriptomics, and what this did is that in this experiment they collected cells that were Lin negative, SCA1 negative, and c-KIT positive, and then they immediately sequenced the RNA in those, and all the mRNAs that your cell makes at a given time is called the transcriptome. So they did single-cell transcriptomics, so they took hundreds of different cells, singled the mRNA from each of those. So then you come up with the profile. Gene A is expressed 20 times, gene B is expressed 5 times, gene C is expressed 100 times. You end up with these profiles, and then you can say, well, is the population, or are these CMPs all the same? In that case, your profiles from one cell to the next will look identical. But what they found is that when they stacked up these transcriptomes against each other, that the cells sorted out into subpopulations. And so the conclusion from these 3 very differently designed experiments is that your common myeloid progenitor is not a homogeneous population. It's actually a bunch of different cell types, and that this is insufficient. Using this scheme is insufficient to be able to tell whether a cell has started to differentiate down a different pathway. So what the new bracket looks like after the other one was busted apart is this. You have your long-term hematopoietic stem cells, then short-term hematopoietic stem cells, multipotent progenitors. So compared to the last figure, these are all the same. Then, from the multipotent progenitor, you have differentiation occur into all these different intermediate forms. So INT on this chart stands for intermediate, and they're not terribly well defined at this point. But you have all these different intermediates, and they haven't really studied the common lymphocyte progenitor, so we'll hold on to that for a second. But the main idea is that you have all these intermediates, and then they end up at different sorts of differentiated cells. So what we had called common myeloid progenitors before actually was heterogeneous group of cells that were intermediates that were already leading down to the pathways for all these different sorts of mature white blood cells, or red blood cells in the case of the erythrocytes. So this looks very different than the textbook model, and so assuming that this model holds, that those three are consistent with each other and that other groups are able to repeat this experiment, we're going to have to rewrite the textbook. And so I just want to leave you with this thought. It's a quote by Carl Sagan. It says, "In science, it often happens that scientists say, you know, that's a good argument. My position is mistaken." And then they would actually change their minds, and you never hear that old view from them again. They really do it. That's from Carl Sagan. And so that's what I like about science, compared to a lot of things that we have in this world, is that science is fairly intellectually honest. And if you have evidence to say something is true, you believe it. If more evidence comes along and it says, well, that's not quite true, it has to be revised to this slightly different way, then you believe it. So I imagine the textbooks to be honest and go from this sort of a differentiation to this sort of a differentiation profile. It might take a couple of years for the textbooks to catch up, but I bet they'll do it. Talk to you later.